genetic manipulation of pest and host plant resistance, including biotech-developed crops like GM corn, potato, soybean, etc. Genetic manipulation is a process where the gene for a particular character is introduced inside the chromosome of a cell. When the gene for a particular character is introduced in a plant cell, a transgenic plant is produced. These transgenic plant exhibit characters governed by the newly introduced gene. It helps in improving crop variety. It ensures food security and insect-resistant crops. It also improves the quality and yield of crops. Genetic engineering or genetic modification. The direct manipulation of an organism's genome using biotechnology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. Genetically modified crops, GMCs, GM crops, or biotech crops, are plants used in agriculture, the DNA of which has been modified using genetic engineering techniques. In most cases, the aim is to introduce a new trait to plant which does not occur naturally in a species. And here's the examples in food crops. Resistance to certain pests, diseases, or environmental conditions. Reduction of spoilage. Resistance to chemical treatments, for example, is the resistance to a herbicide. Improving the nutrient profile of a crop. Plant tissue culture and deliberate mutations have enabled humans to alter the makeup of plant genomes, a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of known compounds. And now here's the techniques. The production of exact copies of plants that produce particularly good flowers, fruits, or have other desirable traits, and to quickly produce mature plants. The production of multiples of plants in the absence of seeds or necessary pollinators to produce seeds. The regeneration of whole plants from plant cells that have been genetically modified. The production of plants in sterile containers that allows them to be moved with greatly reduced chances of transmitting diseases, pests, and pathogens. The production of plants from seeds that otherwise have very low chances of germinating and growing. For example, orchids and nepenthes. To clean particular plants of viral and other infections, and to quickly multiply these plants as clean stock for horticulture and agriculture. And last is the first genetically modified crop plant was produced in 1982, an antibiotic resistant tobacco plant. For the application, a plant breeder may use tissue culture to screen cells rather than plants for advantageous characters, like for example herbicide resistance or tolerance. Production of identical sterile hybrid species can be obtained. Plant Genetic Systems or the PGS, best known for its work in the development of insect-resistant transgenic plants. The focus of its activities is the genetic engineering of plants. First company to genetically engineer insect resistance or the tobacco plants by incorporating genes that produce insecticidal proteins from Bacillus thuringiensis. DNA transfers naturally between organisms. The instruction of foreign germplasm into crops has been achieved by traditional crop breeders by overcoming species barriers. A hybrid cereal grain was created in 1875 by crossing wheat and rye. Since the important traits including dwarfing genes and rust resistance have been introduced, plant tissue culture and deliberate mutations have enabled humans to alter the makeup of plants' genome. The first genetically modified crop plant was produced in 1982, an antibiotic-resistant tobacco plant. The first field trials occurred in France and the USA in 1986, when tobacco plants were engineered for herbicide resistance. In 1987, Plant Genetic Systems, founded by Mark van Montague and Jeff Schell, was the first company to genetically engineer insect-resistant tobacco plants by incorporating genes that produce insecticidal proteins from the Bacillus thuringiensis. Genetically engineered crops have genes added or removed using genetic engineering techniques, originally including gene gun, also known as the biolistic, shoot a direct high-energy particles or radiation against target genes into plant cells. It is the common method. DNA is bound to tiny particles of gold or tungsten, which are subsequently shot into plant tissue or single plant cells under high pressure. And the electroporation, it is used when the plant tissue does not contain cell walls. In this technique, the DNA enters the plant cells miniature pores, which are temporarily caused by electric pulses. The microinjection directly injects the gene into the DNA. The Agrobacterium tumefaciens. Agrobacteria are natural plant parasites, and their natural ability to transfer genes provides another engineering method. To create a suitable environment for themselves, these agrobacteria insert their genes into plant hosts, resulting in a proliferation of modified plant cells near the soil level. The CRISPR clustered regularly interspaced short palindromic repeats are segments of prokaryotic DNA containing short repetitions of base sequences. It has enormous potential application, including altering the germline of human, animals, and other organisms, and manipulating the genes. And last is the TALEN, transcription activator, like effector nucleus, has tail proteins that compose of central domain responsible for DNA binding or a nuclear localization signal, and a domain that activates the target gene transcription. The capability of this protein to bind to DNA was first described in. And also there are types of modifications. First is the transgenic plants have genes inserted into them that are derived from another species. The inserted genes can come from species within the same kingdom or the plant to plant. Second is cisgenic. Plants are made using genes found within the same species or closely related one, where conventional plant breeding occur. And third is the subgenic. In 2014, Chinese researcher Gao Kaixia filed patents on the creation of a strain of wheat that is resistant to powdery mildew. The strain lacks genes that encode proteins that repress defenses against mildew. Gao used gene editing tools without adding or changing any other genes. No field trials were immediately planned. Yields increased to 9% for herbicide tolerance and 25% for insect resistance. Farmers who adopted GM crops made 69% higher profits than those who did not. The review found that the GM crops help farmers in developing countries, increasing yields by 14 percentage points. GM crops grown today or under development have been modified with various traits. And here's the traits. Improved shelf life, disease resistance, stress resistance, herbicide resistance, production of useful goods such as biofuel or drugs, ability to absorb toxins and for use in bioremediation. And here's the genetically modified crops. Here's the illustration. And the pest resistance, the insects from tobacco, corn, rice, viruses from papaya ring spot virus, from the potato leaf roll virus, and the corn is maize dwarf mosaic virus. And let's move on the host plant resistance. The host plant resistance is anything inherited characteristic of a host that lessens the effect of attack. The plant species which are fed upon by an insect are called the host plants. The inability of insect to attack a non-host plant is termed the immunity. A host plant which shows lesser damage is called the resistant, and a host plant showing more damage is called the susceptible. According to Snelling 1941, plant resistance to insects is a quality that enables a plant to avoid, tolerate, or recover from the effect of heavy position or feeding that would cause damage to other genotypes of the some species under similar environmental condition. And according to Painter 1957, defined plant resistance as the relative amount of heritable qualities possessed by the plant that influence the ultimate degree of damage done by the insects. Also according naman kay Koga 1994, resistance to insect is the inheritable property that enables a plant to restrain the growth of insect populations or to recover from injury caused by the populations. And here's the characteristic of the resistance. There are 4 characteristics of resistance. First is the heritable, the relative, measurable, and variable.